Welcome friends to this lecture 4 of week 11 of soil science and technology, and we will be starting to this we will be starting this lecture by describing the basics of diffuse reflectance spectroscopy. In the last lecture we stopped here. We talked about the difference between specular reflection as well as the diffuse reflection, and let us talk about let us go ahead and see the basics of this near infrared spectroscopy. So, we know now right now what is the difference between the diffuse reflection and specular reflection. We talked about the visible near infrared diffuse reflectance spectroscopy. Now, let us see what is the basics of near infrared spectroscopy. So, in classical physics considers the atom as particles with a given mass in the infrared absorption process, and the vibration of diatomic molecules described as follows. For example, here the diatomic molecules HCl, and you can see there are two types of forces. You can see one is stretch and is compressed. It is just like a spring. So, it basically shows the mechanical model of a vibrating atomic molecule. So, when a molecule like this absorbs some infrared radiation, it shows 6 different types of stretching and bending. So, these are the 6 different types of stretching and bending, as you can see here: asymmetrical stretching, symmetrical stretching, scissoring, rocking, twisting, wagging. So, as a result of that, and as a result of that, there is some spectral absorption and some spectral signatures. So, there are different regions, as you can see: near infrared region, mid infrared region and far infrared region, and origin of the absorption in all these regions varies. For example, in case of mid infrared region, you will see that fundamental of molecular vibrations you can see. In case of far infrared region, you will see that molecular rotation, it is responsible for molecular rotation, or in other words, in the far infrared region absorption will occur due to molecular rotation. In the mid infrared region, absorption will occur due to the fundamental molecular vibration, and in the near infrared region you will see overtones and combination bands of fundamental molecular vibrations. So, that is how you see these different types of spectral signatures. So, what are the spectral signatures? You see here we have combined the soil absorption soil spectra of these 3 ranges. For example, this is the visible range, this is the near infrared range, and this is the mid infrared range. Now, in the mid infrared range, these are basically the fundamental absorption features which basically occurs from absorbing the near infrared rays by different organic molecules or groups, and they produce different types of further also some overtones and combination bands, and these are basically seen in this near infrared range. So, it is basically a combined visible near infrared and mid infrared spectrum of a soil. And you can see these absorption features and reflectance features are very much we can see that, and these absorption basically shows basically reflects these absorption features basically reflects the chemical composition of the soil itself, because this absorption actually happens from different I would say from different chemicals or organic chemicals which are present inside in the soil. So, let us see some equipments for VisNIR-DRS spectrodiometer. So, you can see there are 2 equipments. One is called AgriSpec-DRS made by Analytical Spectral Devices of USA, and this is Spectral Evolution 3500-DRS. So, it is actually PSR 3500. So, please correct it. It is PSR 3500 or so these are 2 important instruments nowadays available for VisNIR-DRS measurement. So, let us go ahead and see what is the internal config of these 2 instruments. So, let us see how it works. So, if you see the internal config of these instruments, obviously this is a sample, this is a starting point, and obviously this is an handheld probe. which has a source. So, basically it is shining a halogen white light source, and it is some amount of light getting reflected back from this surface due to diffuse reflection, and this reflected radiation goes through this fiber optic cable to a holographic grating, which basically further divides these into three different regions. One is called the visible to near infrared region, which varies from 350 to 1000 nanometer. Then short wave infrared 1 region, which varies from 1000 to 1800 nanometer region, and then short wave infrared 2, that is 1800 to 2500 nanometer region. So, this VisNIR region basically then goes from this separation filter, which contains 512 silicon photodiode array, which has a which also got which has the 512 silicon photodiode array, and this short wave infrared one is passed through this indium gallium arsenide detector, which has 600 channels, and this short wave infrared 2 also goes through this another detector, that is InGaAs detector with 600 channels. Ultimately, it converts incident photons to electrons or photocurrent, and it ultimately produces the voltages or digital signal, and which is further measured by this control software. So, this is how this instrument basically works. So, another important thing is that for each and every measurement, we after a couple of measurement we have to standardize this instrument. So, for standardizing this instrument, we need a white reference panel, and we call it spectral panel, and this spectral panel, we assume that it has got 99% reflectance. So, it basically requires for optimizing the spectrometer, and it requires for white referencing the spectrometer, that is simplifies the reflectance. So, reflectance, remember, is a function of lambda, that is the wavelength, and it removes the ambient temperature effect also, and also removes the humidity effect, and optimize when we change from one probe to another probe. So, there are different types of probe attachment. There is some handle probe, there is some mug light which you can set over the desktop, and then you can scan it from below, scan a soil sample from below. So, that also can be done by this and when you change the probes, you have to optimize the instrument by using this spectral panel. So, let us see what is the difference between near infrared spectroscopic analysis in the past and now, why it is gaining importance. So, you see in the left, it is basically the mid infrared spectra obtained by ATR, and the right one is the near infrared spectra obtained by diffuse reflectance. The difference which you can easily observe, that the number of spectral features is more in case of mid infrared as compared to the near infrared region, which contains some broad absorption features also. So, NIS spectroscopy was neglected by spectroscopists who for long time could not find any additional attractive information, and in that spectral region, which was occupied by broad superimposed and weak absorption bands. So, this is the reason, that is why the NIR based technologies were not so popular previously. So, you can see it was difficult to interpret these broad absorption features. However, due to the advancement in these three to four different fonts, you can see there is an advancement in the optical fibers, there is an advancement in computing power, there is an advancement in chemometrics, there is an advantage of interest in process analysis. So, as a result of these advancements, nowadays we can use this NIS spectrometer for better management or better prediction or better analysis of any particular soil property. Now, what is chemometric? Chemometric is a specialized data analysis where we measure the concentration of a particular element or chemical using some multivariate techniques or statistical means. So, that is called chemometrics. So, there are several methods of chemometrics. Generally, we use different types of machine learning algorithms for predicting as a chemometric tool, like partial least squares regression, then principal component regression, and so on and so forth. So, if we go ahead and see what these spectral features basically look like. So, you see here, these are the spectral preprocessing. These is actually the spectral signature of a raw soil sample. So, basically what we do, we generally take 4 scan or 5 scan per sample by rotating the sample, because we want to take a homogenized measurement of the scan. We want to take, we want to nullify, or we want to we want to offset the heterogeneity of the soil, because soil is heterogeneous. So, if we take several measurement and then make an average, then it will produce us more homogeneous results or more average results. So, that is why you can see here we are taking 4 raw scan per sample, and after that we produce, using some methods, we produce different one average spectra. And this is a typical reflectance spectra, visible near infrared reflectance spectra of a soil, which you can show some broad absorption features. Obviously, these broad absorption features are represented, these absorption features occur from the moisture or the water molecule which is present inside the soil. So, so, once we get this thing, then we go for the spectral pre-processing. Why? Because you can see, if you go to the last slide, you will see these absorption features are quite broad, and it is difficult to identify the small absorption features or very specific absorption features which are responsible, or which are basically arising from absorption by different components which are present into the soil. So, that is why we require different types of spectral pre-processing. So, you can see here we are doing different types of spectral pre-processing. This is called, these are some common spectral pre-processing. We call it first derivative of reflectance spectra, and this is called second derivative of reflectance spectra. So, in the first derivative of reflectance spectra, we generally take the first derivative of the reflectance values as in comparison to the wavelengths, and in the second derivative we are taking second derivative of the reflectance values in comparison to the wavelength. So, basically what are these? These are basically the reflectance values, individual reflectance values which we have recorded for each of these wavelengths, starting from 350 to 2500 nanometer. For each of these wavelengths, we have basically recorded the their reflectance values, and when we are plotting these reflectance values, these are called the spectrum. So, this spectrum contains these broad absorption features, and to remove these or to further get higher resolution of the spectral features, we are doing different types of spectral pre-processing. Just you can see here we are doing a first derivative and second derivative. Now, the question comes, why derivative spectra? Obviously, the derivative is basically change in reflectance over a bandwidth, which is denoted by delta lambda, where delta lambda is equal to lambda j minus lambda I, where lambda j greater than lambda I. So, basically we take the nth derivative on the basis of this lambda, in comparison to the lambda or reflectance values. And you can see this is an original reflectance feature, and from this original reflectance feature we are taking different types of derivative. First derivative, this is obviously this is the second derivative, this is third derivative, and then again further higher order derivative like fourth derivative. So, you can see different derivatives you are taking. So, what is the reason behind that? Obviously, there are a couple of reasons. First of all, increasing resolution, then background elimination. You can see there are several background features which we can eliminate through by taking the higher derivatives, and also we can take some noise filtration. For example, when we are taking the spectral scan in the field, there may be, or in the lab, there may be some ambient light source, and from those ambient light they can come and they can they can create some interferences. So, this type of interferences and can be nullified by using some spectral pre-processing. So, obviously again also you can see higher resolution. This is a broad reflectance spectrum or absorbance spectrum, which is basically which are basically masking these two important spectral features. So, when you are going from this original absorbance to higher order derivatives, we can see these two analytes can be separate with similar lambda max values. So, by increasing the basically you can see how we are increasing the resolution to get more information about the more analytes which are present in the soil. Similarly, you can see this broad absorption feature is suppressing these small absorption features. So, when we are going from original absorptions to first derivative and second derivative, how these are getting separated, and then how we are getting much more important information. So, these are some reason that's why we use the spectral pre-processing. Spectral resolution is also very important. The spectral resolution, suppose this is peak spectral peak, so obviously this difference is called the spectral resolution, and this is the 50% of the peak height. So, if this is the total peak height, the 50% of the peak height we call it full width half maximum, or full width, it stands for full width half of maximum. So, it is a half of maximum. So, that shows the spectral resolution. Higher the resolution, that means you can higher the resolution means lower the distance, or in other words, you can better resolute the minor spectral features using the spectrodiometer, and these consecutive points are basically shows the sampling interval. So, this is a representation of spectral resolution in the spectral curve. So, again the idea is once we have gathered this database of spectra, for example, we have thousands of samples and we can gather the thousands of spectra for those thousands of samples, and once we gather the thousands of sample spectra from this, we will create a database, and from this database we will do some spectral pre-processing, and from this spectral pre-processing we will be transformed the data, and from using the transformed data we will create some predictive modeling, and from this predictive modeling we will do some interpretation and rule formation, and that is why we use this method extensively nowadays for predicting different soil properties. And we will see that in the next topics, where we discuss about the diffuse reflectance spectroscopy for application for soil. So, let us see some specific examples of diffuse reflectance spectroscopy. You can see here Weiser et al. In 2007, they have used this diffuse reflectance spectroscopy and they have predicted the soil clay. So, you can use that visible near infrared diffuse reflectance spectrometer for predicting the soil clay, and they have got an R square values of 0.92, 0.84 and 0.92, 0.75 and 0.83 for different conditions. Not only they got good results, but also they have identified what are the important wavelengths which are responsible for producing these high prediction results. So, you can see for different conditions like air dried ground condition, field moist in situ condition, air dried in situ condition, all these conditions they have identified these important spectral wavelengths which are responsible for producing high accuracy. So, also by using their model they have seen the good correlation or good conformity between lab clay content as well as the VisNIR predicted clay content. So, you can see soil clay can be predicted by using this technique. Another important aspect of soil is organic carbon and inorganic carbon, and these scientists Morgan et al. They have proved that you can use this visible near infrared spectroscopy, diffuse reflectance spectroscopy to produce the high to produce the quantification, or you can predict both organic carbon and inorganic carbon. You can see organic carbon can be predicted with an accuracy R square values of 0.77, that means 77% accuracy, and in case of organic carbon or soil inorganic carbon, there you can predict it with an R square values of 0.85. So, that shows that this instrument can be used for predicting soil organic carbon and soil inorganic carbon without any consumables or without any chemical used. Also, Zhu et al. In 2010 they have proved that you can use this technology for measuring surface soil moisture. Now, surface soil moisture is generally you can calculate using some gravimetric measurement. This is a gravimetric measurement, and you can convert it to volumetric content of water, volumetric water content by multiplying with the bulk density ratio, and you can see that this the water content can be predicted, the surface soil water content can be predicted by using the scan results from this diffuse reflectance spectroscopy. So, also although it is, we have although the scientists have produced this high correlation between the moisture content and this diffuse reflectance spectra, these moisture content sometimes produce problem for producing higher prediction accuracy, because most of the time this moisture, due to the absorbance of near infrared radiation by moisture, these the features, the spectral features which are the spectral features coming from other soil components are masked, and as a result of that there is some suppression, and as a result of that there is reduction of accuracy of the modeling. So, scientists have used a new technique called EPO, or external parameter orthogonalization technique, for removing the effect of moisture. So, nowadays you do not have to worry about the moisture content in the soil, you can directly take the scan in the field and you can use some mathematical techniques to remove the moisture impact by this EPO method, or external parameter orthogonalization technique, and by using this technique you can directly predict organic carbon as well as clay without any moisture influence. So, this is the advancement of research in this near infrared spectroscopy or visible near infrared diffuse reflectance spectroscopy, and also some Indian application I would like to show you. You can see here our group has used this technology for measurement of arsenic content in landfill site soil. So, we use this technology for measurement of soil arsenic concentration. So, we went to a landfill area where municipal land waste, municipal solid waste are dumped for quite a long time, and around this landfill area there has been a huge amount of agricultural lands which grow the leafy vegetables, which accumulate, which has the potential for accumulating these heavy metals which are coming through these municipal solid waste. So, we thought that we should use this DRS technique for predicting the arsenic concentration. So, we went and collected several soil samples from there. As you can see from this is a study area, and we call it this is a landfill site called Dhapa landfill site, situated in Kolkata city of India, and these are water bodies those. So, these are the sampling points. So, we collected the samples and then we scan using those two techniques. One is the mid infrared spectrometer, we call it drift or diffuse reflectance infrared Fourier transform technique. Another is visible near infrared diffuse reflectance spectroscopy, and after scanning these two using these two methods, we also simultaneously measured their properties like total arsenic content, and also not only the total arsenic content, but also we used a specialized technique called sequential extraction to extract the individual pools of arsenic which is present into the soil. So, you can see there are 5 different pools of arsenic which is present into the soil. One is called ionically bound arsenic, then strongly adsorbed arsenic, then arsenic co-precipitated with carbonates, manganese oxide and very amorphous iron oxyhydroxides, arsenic co-precipitated with amorphous iron oxyhydroxide, arsenic associated with organic matter and sulfides, and we measured them individually, and then using some qualitative spectral analysis we tried to identify whether we can identify those individual features or individual pools. So, this is the actually the visible near infrared reflectance spectrum, and this is the mid infrared reflectance spectrum of the collected soil samples. So, you can see we have identified the goethite and hematite which are important iron based minerals, and then we can identify the hydroxyl of clay minerals and organic matter, bound arsenic here, and we also identified the kaolinites, smectite and organic matter, hydroxyl here, and then other features like CWO of metal carboxylates and then calcite and carbonates and all these things. Not only we have done the qualitative analysis, but also we predicted the total arsenic concentration and we got a very high R square values of 0.97. And the beauty of it, you can use this technology for directly mapping into the field also, or directly producing the contamination map also. So, you can see using this model you can use the geostatistical interpolation which we covered in the last lecture. So, geostatistical interpolation can be used to use these model predicted values to produce the arsenic contamination map. So, you can see we are getting arsenic contamination maps, and for that area showing different levels of arsenic contamination starting from low to very high. Okay. Not only that, we also produce this prediction model for individual soil arsenic pools, and it proved that you can predict these individual soil arsenic pools by using this diffuse reflectance spectroscopy, and also we have identified the individual significant wavelengths by this diffuse reflectance spectroscopy. Now, we are also produce the online mapping of soil properties or on-site mapping of soil properties by using some variable indicators. So, variable indicators uses some specialized variables between this 350 to 2500 nanometer. So, this 350 to 2000 nanometer shows the whole range of spectral variables or spectral wavelengths. Now, we are selecting some spectral wavelengths from this whole range of spectral wavelengths, and then we are efficiently utilizing them in using some model to predict particular soil properties. So, you can see here we are producing the spatial variability maps or kriging maps of particular soil property. Actually, we have produced maps of total carbon, and this is a map of sand, and then map of clay, and then map of silt, and then total nitrogen, total carbon, and loss on ignition method of soil organic carbon. So, you can see we have produced all of these maps using these combined variable indicators as well as this geostatistical mapping techniques. Also, we have proved the usefulness for mapping the soil petroleum contamination. Soil petroleum contamination is a serious environmental problem because it produces the neurotoxic effects. It is so we collected several contaminated samples from different areas, and we plotted their their reflectance spectrum. You can see the differences between the reflectance spectrum originated from the soil which has got no hydrocarbon or petroleum, and also from the soil which are having hydrocarbon or petroleum. So, you can see we have collected two different soils, and then we can see the differences in the reflectance pattern. Also, we have identified these red are the basically important significant wavelengths which are responsible for identifying these petroleum hydrocarbons. So, we produced several types of maps and several types of models. So, we saw that we can predict this petroleum hydrocarbon contaminated soil using great accuracy with an R square values of 0.98, you can see here. And not only that, we use this technology directly into the field for mapping a petroleum spilled area. So, you see this is a spilled area where there is a petroleum petroleum oil spill. It is a actually hilly area. We are looking from the top, and actually the oil moved through this area. So, it is an soil area. So, we collected several soil samples from this area. These are the soil sampling points. After collecting the soil sample points, we model them using we scan them using our spectrodiometer, we model them, and this is the spatial variability map. As you can see, obviously the high concentration of the petroleum is concentrated at this area, and it moved through this area. So, that shows the better accuracy of our modeling. And not only that, we also collected the contaminated soil samples from BP Deepwater Horizon oil spill. There is a Deepwater Horizon oil spill in 2010 in United States. So, we went there and we collected several soil samples starting from very clean sand to very highly contaminated sand, and these are basically the petroleum which is mixed with the sand samples. So, we actually collected the sand samples. and this petroleum actually came from the sea, and it actually contaminated the whole land as well as the beaches. So we went there and we collected these samples from the beaches. These are actual samples. After collecting them, we scanned them with our spectroradiometer, and we use the prediction model, and you can see we are getting the R square values of 0.95 to 0.99. So that shows the effectiveness of our technology. And also we gave some on-site prediction for predicting the soil arsenic, soil petroleum contamination. So guys, it is now I hope that you have got some basic overview of this technique, how it works. Obviously, it is not possible to cover all these aspects within this time frame, but I would request you to go ahead and read some literature. This is a burning topic nowadays, and this is the most advanced techniques that soil scientists are using nowadays for different types of soil research, and it can literally carve down all the efforts of soil science, of soil based estimation. So you can do some more advanced research. Those who are pursuing some research in the field of soil science, you can do this research on diffuse reflectance spectroscopy based modeling of different soil properties. So guys, thank you. Let us wrap up here, and then in the last lecture we will be talking about another important technology, that is portable X-ray fluorescence. Thank you.